Hello learners, today we learn about immunoblotting techniques. Blotting techniques is nothing but it is isolation and quantification of specific nucleic acid sequence and the study of organization, intracellular localization, expression and regulations. The blotting techniques describe immobilization of sample nucleic acid on nylon or nitrocellulose membrane. Also, blotting techniques nucleic acid used as a target in a subsequent hybridization experiment. DNA, RNA, and proteins are easily separated by blotting techniques. So, blotting techniques is nothing but it is the isolation and quantification of specific nucleic acid sequence, and also study of organization, intracellular localization, and expression and regulations. Then types of immunoblotting techniques. There are four types of immunoblotting techniques. That is first, that is ELISA. ELISA means nothing but enzyme linked immunosorbent assay. What is the full form of ELISA? E means a enzyme, L means linked, I means immuno, S means sorbent, and A means assay. ELISA means enzyme linked immunosorbent assay is widely used techniques for detection of antigen or antibody. ELISA techniques is widely used for detection of antigen or antibody. Second test, Western blot. It is used to detect protein. Third technique, Northern blot. It is used to detect RNA. And fourth technique, Southern blot. It is used to detect DNA. So these are the four techniques of blotting techniques. So first one, learn about ELISA. ELISA means enzyme linked immunosorbent assay. So what is ELISA? ELISA is a very sensitive immunochemical techniques which is used for detection of antigen or antibodies. Enzyme immunoassay was the first screening test commonly used for identification of HIV. Also, ELISA is an antibody-based method. It is a plate-based assay which is designed to quantitatively detect specific antigen, proteins, peptides, hormones, or antibody in a sample. Principle of ELISA. We learn about principle. Principle is based on the formation of antigen or antibody reaction. Enzymatic chemical reaction in which catalyze the formation of colored or chromogenic product from colorless substrate. Enzymatic chemical reaction in which catalyze the formation of colored or chromogenic product from colorless substrate. Chromo means color. Signal detection and quantification. Detection and measurement of color intensity of the color product which are generated by the enzyme added in substrate qualitatively or quantitatively. This read by using ELISA plate reader. ELISA microplate reader with 96 well is used, which is well coated with antibodies. Their antibodies may be directly labeled with signal generating enzyme. This signal observed is proportional to the amount of antigen present in the sample. Next, types of ELISA. On the basis of binding of structure between the antibody and antigen, there have three types of ELISA. First, that is indirect ELISA. Second one, that is sandwich ELISA, and third one is that is competitive ELISA. So first, upon indirect ELISA is used to detect presence of antibody present in the sample. Indirect ELISA used to detect presence of antibody. Second one, sandwich ELISA used to detect specific antigen present in sample, and third one, competitive ELISA is used to measure the concentration of an antigen in the sample. Application of ELISA techniques. ELISA can be used for determination of serum antibody concentration in virus tests such as HIV, Nile virus test. First application, that is determination of serum antibody concentration. Second, it is also used in the food industry for detection potential food allergens that cause allergy. It is used in vitro diagnostic in medical laboratories. In vitro means outside the body. It is used in vitro diagnostic test in medical laboratories. And fourth one, detection of mycobacterium antibodies in tuberculosis. And fifth, detection of bird flu. So these are the applications of ELISA. First application, the determination of serum antibody concentration in virus tests such as HIV, Nile virus test. Second one, it is also used in the food industry for detection of food allergens. It is used in vitro diagnostics in medical laboratories, detection of mycobacterium antibodies in tuberculosis, detection of bird flu, etc. Next technique, that is Western blotting techniques. Western blotting techniques is also known as immunoblotting or protein blotting. Is a rapid and sensitive method which is used to detect presence of specific protein in a complex mixture extracted from cells. Western blotting techniques, it detect specific protein. You have to remember specific protein in a complex mixture extracted from cells. The separation of protein mixture by size using sodium dockyl sulfate polyacrylamide gel electrophoresis SDS page. The separation of protein mixture by using sodium dockyl sulfate polyacrylamide gel electrophoresis SDS page. The proteins in the gel transfer to the solid support, i.e. nitrocellulose membrane or nylon using electric current. A technique for detecting specific proteins separated by electrophoresis by use of labeled antibodies. So I have to remember Western blotting techniques used to detect specific proteins in a complex mixture extracted from cells. You have to remember specific proteins detect Western blotting. Next one technique, that is Southern blot. The Southern blotting is a central technique used in a molecular biology for detection of specific DNA sequence in DNA sample. Southern blotting, it can detect specific DNA sequence in DNA sample. Southern blotting is a method which is used to transfer DNA from agar gel to membrane where the DNA can be eventually probed for specific sequence. The method is named after its inventor, British biologist Edwin Miller and Southern, who developed the method in Edinburgh, Scotland in the 1975. In this method, the DNA fragments are separated on the basis of size and charge during electrophoresis. The Southern blotting techniques is to detect a specific DNA sequence in DNA sample. Principle of Southern techniques, that is basic principle of method of is hybridization. Hybridization is a process of base pairing between complementary signal stranded polynucleotide from two different sources under the appropriate conditions of temperature and ionic strength. Application: to identify specific DNA in a DNA sample, to isolate desired DNA for construction of rDNA, identify mutations, deletions and gene rearrangements used in the prognosis of cancer and prenatal diagnosis of genetic diseases. First application, to identify specific DNA in a DNA sample, to isolate desired DNA for construction of rDNA, identify mutations, deletions and gene rearrangements used in prognosis of cancer and prenatal diagnosis of genetic disease. So what do you learn in today's section? Immunoblotting techniques, then different techniques of immunoblotting techniques. There are 4 techniques. Then blotting techniques is used to isolation and quantification of specific nucleic acid sequence and the study of organization, intracellular localization and expression and regulations. Types of blotting techniques, there are 4 techniques, that is ELISA. First, enzyme linked immunosorbent assay is widely used to techniques for detection of antigen or antibodies. Western blot, it is used to detect protein. Northern blot, it is used to detect RNA, and Southern blot, it is used to detect DNA. First one, that is ELISA, detect antigen and antibody. Western blot detect specific protein. Northern blot, it is used to detect RNA, and Southern blot, it is used to detect DNA. Then we learn about ELISA. ELISA is a very sensitive immunochemical techniques which is used for detection of presence of specific proteins which can be antigen or antibodies. This is the first screening test commonly used for identification of HIV. ELISA is a antigen antibody based method. Antibody based methods. It is a plate based assay which is designed to quantitatively detect specific antigen, proteins, peptides, hormones, or an antibody in a sample. Then principle is based on the formation of antigen and antibody reactions. Enzymatic chemical reactions in which catalyze the formation of colored or chromogenic products from colorless substance. Those substances have colorless that form chromogenic. Chromo means color. Signal detections and quantifications, detection and measurement of color intensity, color product which are generated by the enzymes and added substrate qualitatively or quantitatively, read using by ELISA plate reader. ELISA microplate reader with 96 wells is used in which well are coated with antibodies. Their antibodies may be directly labeled with the signal generating enzymes. These signals observed is proportional to the amount of antigen present in sample. Types of ELISA, there are three types of ELISA: indirect, sandwich, and competitive ELISA. Indirect user detect antibody, sandwich ELISA detect antigen, competitive ELISA detect concentration of an antigen sample. Applications: detection of antibody concentration, identification of HIV, Nile virus test, also detection of food industry for detecting potential food allergens. It is used in vitro diagnostics in medical laboratories. Detection of mycobacterium antibodies in tuberculosis, detection of bird flu, etc. Western blotting, it detects specific protein in a complex mixture extracted from cells. It is also known as immunoblotting techniques or protein blotting. Southern blot, it is a central technique used in molecular biology for detection of specific DNA sequence in DNA sample. Application: to identify specific DNA in DNA sample, to isolate desired DNA for construction of rDNA, identify mutations, deletions, and gene rearrangements used in prognosis of cancer and prenatal diagnosis of genetic diseases. So we learn about today's lectures: immunoblotting techniques, various immunoblotting techniques, ELISA, Western blotting, Southern blotting, Northern blotting, its applications, as well as principles. So thank you.